Hello everyone, welcome to ISF College of Pharmacy. As you know that ISF College of Pharmacy, Moga, Punjab, India is conducting ISFCP dialogue series under the aegis of Internal Quality Assurance Cell. As well as ISF College of Pharmacy have done memorandum of understanding with various institutions, organizations and stakeholders. Similarly, we have a joint venture with SCOPE, that is Smriti College of Pharmaceutical Education and Research, Indore, Madhya Pradesh. In that series, we are conducting this ISFCP dialogue series lecture. Every day, eminent pharmaceutical scientist, key person is coming to this platform and delivering their thought process. Today, we have very eminent speaker with us. I would like to welcome our today's speaker, Dr. Rajiv Saxena, sir. Welcome, sir.
Thank you, sir.
And I would like to introduce Rajiv sir profile to our audience. Dr. Rajiv Saxena sir is working as professor at Smriti College of Pharmaceutical Sciences. He had received University Gold Medal in his post-graduation and did PhD in Pharmaceutical Sciences from the prestigious B.R. Nahata College of Pharmacy, Mandsaur University, Madhya Pradesh. He is having more than 2 decades of experience to his teaching and academic and research credits in B Pharm, M Pharm, PhD. He has supervised many research scholars during his tenure. He is specialized in pharmaceutical biotechnology, microbiology, biochemistry. He has guided more than 2 dozens of PG students and more than 4 dozens of UG students for their academic research. He has published more than 25 research papers in national as well as international journals of high repute. He is an author of 5 books from reputed publication house, including CRC Press, CBC Publishers, etc. He had published 5 international chapters in Royal Society of Chemistry, Academic Press, Elsewhere, IGI Global, etc. He is awarded with Best Teacher Award, Young Researcher Award, Excellence in Teaching Award, Inspirational Associate Professor Award, and more than 10 Best Research Paper Presentation Awards. His area of research includes molecular biology, plant tissue culture, and electric technology. He is a live member of IPG and IPA. He had published 12 Indian patents in the field of formulation and development. 9 design patents have been also granted. As an academic representative, he has signed MOU with many pharmaceutical industries like NEO Drugs, Safi Healthcare Limited, and Atal Incubation Center. He has also worked as co-PI in research project sponsored by Madhya Pradesh Council of Science and Technology, Bhopal. He is Innovation Ambassador of MHRD-IIC. Overall, Dr. Saxena is a good human being, a thorough teacher, a good academician, administrator, and researcher. With these words, I would like to welcome to you ISF College of Pharmacy, sir. And please start your proceedings, sir. Welcome, sir.
Thank you, sir. A very pleasant morning to one and all present here. Thank you, sir, for this lovely introduction. I also extend my heartfelt thanks to respected Professor G.D. Gupta, sir, and Dr. Saurabh Kwasi, sir, for providing this wonderful platform to share the idea as a part of MOU activity. So today I will be discussing about a very potential but less explored research idea that can be implemented with the help of an interdisciplinary research, that is Elicitor Technology, a novel approach to enhance plant secondary metabolite. Sir, please let me allow to share my presentation.
Yes, please, sir. Share.
Yes. So plant-derived medicines plays a very important role in the treatment and management of various dreadful diseases. Now, if we'll talk about the popularity of these medicines are very popular in the developing countries, and people are using this medicine even in the developed countries to strive good health. But because of the overpopularity of these medicine, the natural resources are getting exploited. And because of the popularity of this medicine, many new herbal industries are nowadays introduced. And every pharma industry who is engaged in the production of the herbal products or maybe the research organization, they are completely dependent upon the natural resources for the continuous supply of this raw material. But it has been observed because of the overexploitation and because of certain external factors including ever-changing climatic condition, global warming, and pollution, they are adversely affecting the rich flora and fauna of the country. The overall impact of this situation is many rare varieties of the medicinal plants nowadays they are getting extinct. So, every branch of science is giving its contribution in order to preserve this rich flora and fauna, so as the case with biotechnology. Various biotechnological approaches are nowadays available that can contribute in the enhancement of the plant secondary metabolite, maybe by the use of the tissue culture. So different approaches are now being adopted by the biotechnology, including the use of the precursor with the help of the biotransformation reaction to the change in the climatic condition of growth, maybe by the change in the media constituent, and also by the use of the elicitor. So today I am going to emphasize more on the elicitor technology. As we all aware that elicitors are actually the agents, or these are actually the molecule that have the capability to trigger the immune response. Now, because of the triggering of the immune response in the plant, the different varieties of the plant secondary metabolites are generated with the help of different biosynthetic pathway. As we know that in the earth, every organism is having a well-defined immune mechanism. If we talk about our body, whenever we encounter an antigen, then the immune system elicitation in our body takes place. And as a result of which, different defensive molecules like antibodies, T cells elicitation, etc. takes place. And the end of these particular elicitation is the incoming pathogen can be invaded and can be nullified from the body. So similarly, if we talk about the plants are also having a well-defined immune system. So whenever a plant is attacked by a pathogen, a series of signaling cascade will be initiated, and that results in the formation of various defensive molecules, including your secondary metabolites, your phytoalexins, various pathogen related proteins. These are all will be generated by the cell, and the end of that is that the plant can achieve the immunity against the pathogen. So here when we are talking about the elicitor technology, so here basically we are trying to mimic the action of the pathogen with the help of the elicitor. So under the artificial condition, when we are going for the application of the elicitor, the plant cell recognize it as a foreign molecule, and again the similar type of the signaling cascade elicitation will takes place, and that result in the generation of various plant secondary metabolite. And also it has been observed in many times that with the help of the elicitor, many new secondary metabolites which are categorized as a phytoalexins can also be generated by the plant cell. So in short, we can say that here we are artificially creating the stress condition in the plant cell with the help of an elicitor, so that the different varieties of the plant secondary metabolite can be generated, can be enhanced depending upon the concentration of the chosen elicitor. If we classify the elicitor, then broadly, these elicitors are classified into two categories as the abiotic elicitors and biotic elicitors. The abiotic elicitors are again divided into three categories as the physical, chemical, and hormonal abiotic elicitors. And the biotic elicitors examples includes various polysaccharides, yeast extracts, fungal bodies, and various bacterial cells. So again, coming to the mechanism of the action of these elicitor, we can explain it by taking a simple example of salicylic acid, role of salicylic acid in the flavonoid production. So if we'll talk about a plant body, so inside a plant body, a gene is available which is called as a PAL gene. The full form of the PAL is phenylalanine ammonium lyase enzyme gene. So this gene is basically responsible for the production of an enzyme that is phenylalanine ammonium lyase enzyme. But this enzyme or this particular gene is actually elicited by the presence of the salicylic acid. So in short, we can say that because of the upregulation of the PEL gene, the involvement of salicylic acid is available. So if we'll talk about a plant body, then in a plant body, the salicylic acid pathway is available. And whenever a plant undergoes a stress condition, maybe because of the incoming pathogen, maybe because of certain drought situation, then as a result of which the salicylic acid is generated by the plant. And because of the generation of the salicylic acid, the upregulation of the PAL gene takes place. And once the PAL gene is upregulated, the conversion of the phenylalanine into transenamic acid takes place. And once the transenamic acid is generated in the plant cell, again a various biotransformation biochemical pathways are activated in the plant that ultimately result in the formation of the secondary metabolite, for example, epigenin. So we can say that, in short, we can say that the salicylic acid is here actually responsible for the upregulation of the PAL gene that ultimately result in the formation of the secondary metabolite, for example, the epigenin. So if we talk about the different plant secondary metabolite categories, then especially for combating the incoming pathogen, various categories of the phenolic and flavonoidal compounds are responsible. So if we see the results of various researches that has been done in this particular field, then we'll find that various scientists have worked for the production of different varieties of the plant secondary metabolite like chiconin, the anthocyanins, the rosmarinic acid, various Taxol. These type of the products are generated by the use of different varieties of the biotic elicitor, that is maybe for example by the use of the chitosan, by the use of the fungal elicitor, by the use of salicylic acid, jasmonic acid and methyl jasmonate. So here in short we can say that the elicitors are the compounds when we artificially generate the stress condition into the plant cell, then these elicitors are actually responsible for the generation of different varieties of the plant secondary metabolite. So in short, we can say that suppose if we are having a pharmaceutical industry that is ultimately dependent upon the regular supply of the phytochemical, then these phytochemicals, we are not dependent upon the natural resources, but rather with the help of the elicitor, we can generate these secondary metabolites in our laboratory. So now the question comes, like if we have to start our research in this particular elicitor technology field, so then how we can start this research? So here I am giving a very small example of the research that recently we have completed in our organization. So the research was started with the construction of the greenhouse. So here we are constructing the greenhouse because we want to create a controlled environmental condition so that the acclimatization of the plant can take place and the growth of the plant can be done in an optimized manner. So after the construction of the greenhouse, we need to select a particular plant from which we need to go for the enhancement of the secondary metabolite. So if we talk about the research problem, if we are talking about the research problems which are selected, especially in the branch of pharmacognosy or pharmacology, then the first question is which plant we need to select. So usually the students go for the study of the plant that is less explored and the plant on which that particular pharmacological activity has not yet been performed. But if we talk about the elicitor technology, there is no such hurdles regarding the selection of the plant. We can select any plant which is having the medicinal importance, but even if we go for the selection of the plant which is at the edge of extinction, that would be a better choice. So once the plant is selected, we need to sow this plant in the greenhouse that we have already constructed. So after the construction and after the sowing of these plant in the medicinal plant, in the medicinal garden, then what we need to do, we have to wait for around 30 to 40 days till the plant get acclimatized in the greenhouse. Because once we see that the plant is now adopted with the environmental condition of the greenhouse and it is showing very good growth signs, then only we can go for the start of our research with the help of different varieties of the elicitor. So once everything is set, your greenhouse is ready, you have already chosen your plant, you have already sown the plant in the greenhouse, and you have seen that your plant is acclimatized with the environmental condition of the greenhouse. Now we can start our elicitor technology. So from this point, we can divide our research into two areas. Number one is we can do the research in the field condition, and the second important area is we go for the plant tissue culture technology. So but before doing this particular thing, we need to go for the testing of the soil, because we know that in the soil that is available in our medicinal garden may already have the availability of maybe some varieties of the plant pathogen. So in order to assess the effect of a particular elicitor on that particular plant, we need to assess the quality of the soil. So in every state, different soil testing laboratories are actually available, and these soil testing laboratories provide the soil health card. So various governmental organizations provide the soil health card. You just provide that sample to these organizations and they will give you the data regarding the health of your soil, including the presence of the pathogen in the soil. So once the soil testing is also be done, then we can start our plant tissue culture protocol. So the important plant tissue culture protocol that is used in the Elicitor technology is the callus culture. So initially we need to go for the collection of the explant from the healthy plants which are available in the greenhouse, then followed by the surface sterilization of the explant. And after the surface sterilization, the callus culture protocol optimization has to be done in the laboratory. For that, different types of the plant growth regulator are chosen, like for example, various types of the auxins and cytokines, including your indole acetic acid, benzyl aminopurine, in a different concentration has to be incorporated into the morassic and scoob media. As you can see in this particular diagram, this is the research that recently we have completed. So the plant is actually Gardenia gumifera. So we have chosen different concentration of indole acetic acid and benzyl aminopurine, and we have observed the conversion of these explant into the callus. So it takes around 20 to 25 days to convert this explant into the callus body. We know that the callus is an undifferentiated mass of the cell, and it is actually representing the plant cell, and it has a capability to generate various secondary metabolites. So once the callus culturing is completed, we need to convert this callus into the cell suspension form. So in the next step, after the formation of the callus, we need to add this callus body into the broth culture that consists of the broth culture of morassic and scoob media, that is also supplemented with the hormones that we have already utilized for the formation of the callus culture. So it takes around 4 to 5 hours in order to convert the callus into the cell suspension culture. So as discussed earlier, the callus is a very fragile mass of cell. So once you incorporate the callus into this liquid media, and when you put it into the orbital shaker and give the rotations around 110 rotations per minute and a temperature of around 25 degrees centigrade, so maybe in 4 to 5 hours, this fragile callus mass will be shattered in the form of the cell suspension culture. So once the callus is actually converted into the cell suspension culture, then we need to go for the application of the elicitor. So here the protocol, until here, the protocol is very clear. First, we need to go for the acclimatization of the plant into the greenhouse. And whenever we see the healthy signs of the growth of the plant, we need to collect the explant, and that explant is actually converted into the callus mass, and from the callus mass we need to go for the conversion of the callus mass into the cell suspension culture. Now in the next stage we need to go for the addition of the chosen elicitor into the cell suspension culture. But before addition of these elicitor into the cell suspension culture, we need to see what is exactly the growth pattern of your cells, because we know that in case if we talk about the microorganism, then in case of microorganism, initially the lag phase is there, then the logarithmic phase will be achieved when you will find the maximum growth of the microorganism. So similar cases there with the help of the plant cell also, like when you go for the addition of the callus mass into the cell suspension culture, so within 4 hours it will be converted into the cell suspension, but you will not find the growth of the plant cell. So it will take around 10 to 15 days in order to achieve the maximum biomass inside the cell suspension culture. So once we understand the pattern of the growth of the cell in the cell suspension culture, and when we find that what is exactly the time of the logarithmic phase, then only we have to go for the addition of the elicitor. Because we know that whenever the plant cell is in the logarithmic phase, then only the plant will go for the production of the maximum concentration of the plant secondary metabolite. So initially when you go for the cell suspension culture, and at that time if we go for the addition of the elicitor, maybe the concentration of the plant secondary metabolite will not get the maximum. So what we have to do, we have to wait, we have to find out the exact period of time when the maximum biomass is achieved inside the cell suspension culture. So for example, if the maximum biomass is achieved at the 10th day, so maybe you can go for the application of the elicitor on the 8th day, because on the 10th day the maximum biomass conversion will be there and the maximum metabolic activity of the plant cell will be there on the 10th day. So on the 8th day, we'll go for the application of the chosen elicitor. So once the growth pattern is study has been done and we have achieved the day in which we have to go for the addition of the elicitor, then we need to go for the application of the elicitor in various concentration. Actually, when we see the literatures, then we'll find that there is no particular fixed rule of choosing a particular elicitor for the enhancement of a particular plant secondary metabolite. Various varieties of the, as we have discussed in the previous slide, different classes of the elicitors are actually available, and different scientists have used different elicitors for the enhancement of different varieties of the plant secondary metabolite. It means there is no any thumb rule for the selection of a specific elicitor for a specific plant for the generation of a specific secondary metabolite. But here, for example, in my case, we have used the salicylic acid and jasmonic acid because the literature, because our objective was the enhancement of the flavonoid and phenolic compound. So in many papers, we have find like the salicylic acid and jasmonic acid is most commonly used for the enhancement of the flavonoid and phenolic compound. But again, I'm saying there is no particular fixed rule of selection of a particular elicitor. For example, if any student want to start this research, then maybe you can go for the use of 5 to 6 different varieties of the elicitor and try to find out the effect of different elicitors on the concentration of different secondary metabolite of a chosen plant. So it is actually the hit and trial way by which we can go for the optimization and selection of a specific elicitor. And again, we need to go for the optimization of the concentration of the chosen elicitor. Because like, for example, if we talk about the salicylic acid, so people have tried the use of salicylic acid right from 100 millimole to maybe up to 1000 millimolar concentration. Salicylic acid people have tried for from 1 millimole to 10 millimolar concentration. So again, it depends upon different varieties of the elicitor, like which concentration will be more suitable. And it has also observed, like when we go for the increase in the concentration of the elicitor, we'll find that the concentration of the plant secondary metabolite is also enhances. But at a very higher concentration, maybe you will find that there is no significant increase in the concentration of the plant secondary metabolite. So again, you can get all this result when you do the research in the laboratory. Like we need to go for the preparation of the different concentration of the chosen elicitor. We need to go for choosing the different elicitor and trying to find out the defect of these particular elicitor on the plant secondary metabolites. So once we'll go for the application of the chosen elicitor in the cell suspension culture, then again we have to wait for around 24 hours, or maybe we have to wait for 96 hours. Now after 24 hours, we need to go for the harvesting of the plant cell, and even after 96 hours, we need to go for the harvesting of the plant cell, because what we know that now the cell is actually available in the cell suspension form, and at the end of the 24 hours or at the end of the 96 hours, we need to filter out these plant cell, and we need to assess the availability of the plant secondary metabolite in the plant cell also, and even in the filtrate also, because we know that some secondary metabolites, they are generated by the plant and they are available inside the plant only, but some secondary metabolites, they are generated by the plant cell and they are released into the external environment. So after converting the callus mass into the cell suspension culture, after a particular period of time, after the application of the chosen elicitor in a different concentration at various time interval, we need to go for this filtration of the cell mass. And once we filter the cell mass, we have two type of things. One will have a dried cell mass and one will having a filtrate. So we need to assess the availability of the secondary metabolite in both the situation. Maybe the secondary metabolite is available in the cell mass and maybe the secondary metabolite is available in the filtrate. So after getting the filtrate or after getting the cell mass, we need to go for performing the normal extraction process. Maybe we can go for the solvent-solvent extraction in case of the filtrate, or maybe with the help of the cell mass, what we can do, we can dry the cell mass, and from the dried cell mass, again, we can go for performing the extraction process. Maybe you can go for the solvent extraction by the use of different solvent. Like if your target is flavonoid and phenolic compound, maybe your ethyl fraction will get the maximum concentration of the phenolic and flavonoidal compounds. So after performing the solvent-solvent extraction process, we need to judge, we need to quantify, we need to qualitative analysis of the extract, because sometimes it has been observed, like when we perform the experiment, our target is the enhancement of a X phytoconstituent, but the effect of the chosen elicitor on the plant cell, we cannot judge or we cannot assess. Like various phytoalexins are sometimes generated. When we read the literature, we have find like the plant which is normally the phytochemical which is normally not generated by the plant, sometimes because of the stress condition, because of the application of a specific elicitor, some new variety of the secondary metabolite is also generated by the plant cell. So in order to assess which type of secondary metabolite is actually generated by the plant, we need to go for the qualitative and quantitative analysis of the extract, maybe by the use of the TLC, maybe by the use of HPTLC analysis. Then only we are able to assess that which type of secondary metabolite is actually generated by the plant. So this is actually the part of the research when we are actually performing the plant tissue culture protocol for the enhancement of the plant secondary metabolite. But one more type of study that can be planned with this parallel research, we can directly go for the application of the chosen elicitor under the field condition. This type of experiment also we have performed in our laboratory. We have directly spread the elicitor on the plants which are grown in the field condition, and regular application for around 30 to 40 days has been done, and at the end we have collected the leaf, and then the leaves are actually dried, and then normally the extraction, like when we go for the oxalate extraction or similar type of extraction protocol is performed, and again from the extract we have gone for the qualitative and quantitative analysis. So again, in that case also sometimes we can find some significant result, like because it is not always possible to have all the facilities which are required for the plant tissue culture protocol in our laboratory. So if we have to go for the practical application of this elicitor, so for that reason we have tried to apply the elicitor, chosen elicitor, directly into the plants which are grown in the field condition in the greenhouse. And at the end of the study, we have collected the leaves, and then we have dried the leaves, and like whatever we do in the pharmacognosy, various pharmacognostic analysis, qualitative, quantitative analysis, determination of the total flavonoid, determination of the total phenolic content was done, and then we have matched the result that is obtained from the tissue culture protocol and the field condition. And the result that we have got, like definitely it is better to perform the plant tissue culture protocol, because more better result or more significant result are obtained in the plant tissue culture, because everything is in our control. But when we talk about the field condition, maybe certain external factors are also working on it, but it is not like that. Because of the application of the elicitor, no such significant result was observed. Even in case of the plants which are available in the field condition, we have find that there is an increase in the concentration of the flavonoid and phenolic compound, like we have done for the plant that is Gardenia gumifera. So if we have to go for the research in this particular field, then we can go for the research with the help of the plant tissue culture protocol also, and we can go for the research with the help of the field condition plants also. So after the getting the extract, we can also go for clinical correlationship, like whatever the plant secondary metabolite or whatever the extract that we have collected. From that extract, we can go for performing the antimicrobial activity, antioxidant activity, anti-inflammatory activity, etc. can be performed. So here we can find out the clinical correlationship between the different extract. Like for example, if we have chosen 5 different concentration of the elicitor, so we have a 5 different varieties of the extract, and then the clinical correlationship can be established. Like for example, we can perform the antimicrobial activity with the 5 different extracts that are subjected to different concentration of the elicitor. So at the end, we'll find that with the increase in the concentration of the elicitor, more antimicrobial activity, more antioxidant activity, or more anti-inflammatory activity has been observed. And this activity is because of the enhancement of the plant secondary metabolites by the plant cell. So here we can conclude like the Elicitor technology is a promising approach to enhance plant secondary metabolite, and with the help of Elicitor technology, significant industrially important secondary metabolite can be generated under the artificial condition, and these secondary metabolite can have a wider application in contributing to the sustainable agriculture and even to the human health. Thank you so much.
Thank you, sir. Really, it was an interesting talk, and you have covered the dimensions. Really, what you have did, sir, and it is very, very rare cases of such kind of experiments in scientific extempore. And you have wonderfully covered all the prospects of electric technology. And if I talk about from the beginning, it was just like everybody nowadays, herbal technology was very on role in pandemic 19 COVID-19.
Yes.
Certainly, you covered all the parts, sir. And herbs are the life, really, and they are used to treat various disorders. And this elicited technology, biotransformation, its constituents, use of various abiotic, biotic pathways, and the role of it into various productions have significantly touched up to our audience. Definitely, sir, your this talk will motivate the pharmaceutical scientists to spread their knowledge and research dimensions in this field. So thank you so much, sir, for joining this ISFCP dialogue series. And in future also would like to have your expertise in this session. So on behalf of ISFCP IQAC, I would like to thank for your time and candor. You have really given a wonderful talk today. Thank you so much, sir, once
Thank
again.
you so much. Kindly accept my heartfelt thanks. And thank you so much, ISF College, for this wonderful opportunity.
Sincere gratitude, sir. Thank you
See
so
you
much,
soon,
sir.
sir. Thank you, sir.